Welcome everybody to the very last lecture, lecture 40 of ZOO3649 Evolutionary Genetics. Congratulations to all of you who've made it this far and have been concentrating, because that is the key. It's been a pleasure doing these lectures for you, and I really just do hope that you don't miss out any lectures. You download everything, and you watch everyone. And if you don't understand because I say it too fast or whatever, just go back 10 seconds, go back 30 seconds, listen to it again. Listen to it until you understand it, because you're so lucky to have them on video. Unlike all the years before you, they were not so lucky. They had to sit there, busy scribbled notes while the lecturer is talking. You don't have that problem. You have got the whole videos of every single lecture, so you can go back and listen and rewind and rewind and listen again. So when you fail the exam, you failed the exam. Okay? There's no way to explain why you didn't know the content anymore. It is your fault for failing the exam. Okay? So I hope you've taken my advice, and I'm advising every lecture the same thing. All right? Please download everything, and I really hope that you do manage to prepare well for the next test and for the exam. And I can tell you now that if you watched every lecture, you've got every video, and you've listened to my advice and my explanations, you will have absolutely no problem in getting very good marks for this module. Okay? And so let's continue with our very last lecture. It's about conservation units, and this is something that we're going to get into much more during honors, but I'm giving you a taster here in the last lecture of ZOO3649, because this last section of molecular ecology is to take all the things you've learned, the theories, and put it into practice. And you learned a lot about trees, didn't you? And allele frequencies, paraphyly, monophyly, incomplete lineage sorting, gene flow, how to tell gene flow when all you have is a simple tree gene genealogy. All that stuff is up here now. And you are equipped with power and knowledge. And if you're not equipped with that yet, please, I ask you, for your own sake, watch the videos again and again and again. All right. So let's continue our lecture. What are conservation units, and why do we need them? Okay. Well, we would not actually need conservation units if the world was not messed up. Okay. If it was a perfect place where humans did not exploit and were greedy, and we did not destroy everything that we touch, then we wouldn't need conservation units, because conservation would happen without humans needing to do it. Okay? So the problem is that we have messed up the world, and the world is actually in dire straits. And because humans are greedy, 99% of conservation has to do with the greed. Okay? And so when I say greed, I mean politics. It's the same thing. Greedy, politics, they're the same. Politics is just a way of saying individual interests. Okay? It's the opposite of the interests of the country. Politics is just a way of saying individual interests. Okay? And so in conservation, it's individual interests that hold more sway than science, which is why we say conservation is 1% science and 99% politics, because most of the decisions made are not based on the science. They're based on what people want rather than the reality of science. Okay, and so conservation depends on politicians, and politicians depend on money. Politicians don't become politicians because they're nice people. They want to make money, and they want to steal your money. That's why they become politicians. And so when they've got the money, they want to spend it only on themselves. They don't want to spend it on animals and conservation, and they certainly don't want to spend it on their own children's future. They want to spend it on their Ferraris and their pretty girls right now. They have no intention of putting money for the future, for saving species. Okay? There are very few politicians like that. If you know any, you should vote for them. Okay? If you don't, but odds are you don't even know one. I don't, for example, I don't know one. Okay? But therefore, we need to actually define biodiversity in terms of units. Because what we need to do is to convince the politician to spend money to save a species or a population, right? And as I said, politicians think about themselves and not about the environment or the species or the country or the people, only themselves. So you need to bring an argument to the politician that is absolutely hard, strong argument. You can't say, can you give me $1 million to save the rhino? They will say, but why is the rhino worth saving? And then you are scratching your head, you're thinking, Aish, well, I don't know exactly why is it worth saving. Then they will say, well, so why should we give you money, right? So the way to avoid that is to say, dear honorable so-and-so, here I've done a genetic study. Here we have this amount of variation 100 years ago. Now, in today's days, we've got 1% or 10% of that variation remaining. So we've lost, in 100 years, we've lost 90% of our variation. Now, the variation is happening in these places. Most of the variation is in these 3 countries. Please, we need to concentrate our conservation efforts and our money on those 3 countries. Now, that is a better plan, isn't it? Than just saying, can you just give me some money because I want to save the rhinos? Now you are saying, can you please give me money because I want to save what? These rhinos in these countries because of this and this and this reason. This is why they are special. And if we don't do it in the next few years, it's going to be extinct. That's now a good reason. Now he can't avoid you anymore. He can't say, no, now he has to give you the money, right? And that's why we need conservation units, so that you can give the politician a nice package and say, this reason is why we need the money to protect, because of the genetics, okay? Otherwise, forget about getting the money, right? We can't get them to spend money. We can't get them to spend money on just, wishy-washy ideas. They say, spend your money on this thing here. This thing, we define it like this. This is where it exists. This is how much variation it contains. This is why it's important. Okay? So we need conservation units. So the question becomes, should we be conserving species? Firstly, as you all know, there's no way to define a species, right? But if there was, the next question would be, is the biodiversity only the number of species present? Really? So if we count the biodiversity, the number of species, then is that the way we can measure the biodiversity? Are all species equally relevant for biodiversity? Must the biodiversity be determined entirely by our species definition? These are actually questions which are important, very important, because one way of one measurement of biodiversity, don't forget, is genetic diversity. In fact, it's the most basic measurement of biodiversity, okay? And within a species, okay, it is even more difficult, as you know, to define units within a species, because we can't even define a species properly. How are we going to define units within a species? How are we going to break up a population and say, this is big species occurs all over Africa, and we've got a separate population in Kenya and Tanzania, and that population is separate from South Africa and Zimbabwe? We need to look at genes and lots of individuals within each species, okay? And we basically, if we find that the genetics are quite different, then we need to manage them separately, okay? Because their genetics might be quite different because they have local adaptations. And so if we're managing them, we can't just bring a roan antelope from Kenya to South Africa and take a South African roan antelope to Kenya and have it mixed with the Kenyan roan antelope. Why? Because roan antelope in Kenya might have different adaptations to our roan antelope in South Africa. And so if you just move them, do gene flow between things that are not naturally connected, you're going to mess up the selection happening in both those areas, okay? You're going to replace some alleles with your artificial gene flow. You're going to replace some alleles with ones that are not the fittest ones for that area. Okay? So we need to know how different populations are before we can figure out how we can manage them. Okay? So some populations may be on the path to becoming quite different, right? They may show significant adaptive differences to particular niches, right? So for example, black wildebeest and blue wildebeest. Black wildebeest is a high felt specialist, whereas a blue wildebeest is a low felt specialist, right? So significant adaptive differences to local niche, to ecological niches. There can be significant genetic differentiation, okay? So if you can justify that the two populations are different evolutionary lineages, then you can say, well, that means that they're important for conservation because evolution took a long time to make them. Therefore, they are special. It will take very long time to recreate them, right? So we can't afford to lose them, right? Because evolution took a long time to make them. They are significantly different. So phylogeographic studies, like we studied in the previous lectures, phylogeographic studies can help prioritize these areas because phylogeographic studies looks at gene trees for one species across its range, and they look at the patterns of how the gene trees, how the populations are related to each other, okay? And that information can also help to define units for conservation, okay? And the most famous unit and the most significant, if I can use the word unit, for genetic conservation is the ESU, the Evolutionary Significant Unit. Okay, and these are genetically differentiated populations, so two populations that have become different thanks to being isolated in genetic drift, that have a high priority for separate management conservation. So they've become genetically different, they've had no gene flow recently, and so because evolution took a long time to make them different, it will take a long time to replace them if one of them goes extinct. If evolution took 1 million years to produce two populations, then you will need another 1 million years to recreate them if they go extinct. Okay? And we don't have 1 million years. So that's why we need to conserve these, right? Because we don't have the opportunity to get them again. That's how special they are. Okay? So the evolutionary ESU is the most significant unit. So when something is defined as a population or species is defined as an evolutionary significant unit, then it has the maximum priority for separate management and conservation. That's what you say to the politician, put your money here. This is where we need to invest because this is an ESU. Okay? So the ESU is closely related to the idea of subspecies, or the DPS, as the United States Endangered Species Act defines an DPS in exactly the same way, almost exactly the same way as an evolutionary significant unit. Okay? But whatever you want to call them, ESUs, subspecies, DPS, they all merit separate management and have a high priority. Okay? That is because their conservation aims to protect evolutionary processes and adaptive potential. In other words, to make sure diversity is high. Okay? So that if the environment changes and there's a different selection pressure, you have a different fitness landscape, the population can survive. They're not all going to die because why? They're all closely related and they all have the same allele. Okay? Or they have such a small size, they all have the same allele because of genetic drift. They're not going to die for that. We need them to have high diversity, okay? So that they can have a higher potential for survival, okay? And so, so the conservation of genetic variation within a species is very important. And ESU allows you to say, this species can be divided into X number of ESUs, and each of them holds a big piece of the species' total genetic diversity, and therefore they should be conserved. Okay, so an ESU is a way to give a politician some bite-sized chunks of information that they can understand and feel compelled to spend money saving that ESU. Okay, so how do we define an ESU? It's actually quite difficult to define, and because when we say two populations are genetically differentiated, what do we mean by that? How much differentiation? So Craig Moritz in 1994 came up with a seminal paper on conservation units, and he proposed that ESUs can be defined completely by genetics, and they should show significant divergence and reciprocal monophyly for mitochondrial DNA. So in other words, if you have a monophyletic clade of mitochondrial DNA within a species, okay, a monophyletic clade, it means that that population or that clade has been around for long enough for genetic drift to sort the lineages completely. There's no incomplete lineage sorting there because you've got the evolution of monophyly, and monophyly takes a long time to evolve. Okay, and so when you have mitochondrial DNA that shows monophyly reciprocal, so both clades are monophyletic relative to each other, then that is a sign that those two clades have been separated for a very long time. Okay, because if you didn't have monophyly, what would you have? You'd have paraphyly or polyphyly. In other words, either incomplete lineage sorting or gene flow has brought those two clades back into secondary contact. Okay, and that's why you have incomplete lineage sorting, because of gene flow. So if you have monophyly, it tells you no gene flow has happened for a long time, and ILS has been happening also for a long time. Okay, and at nuclear loci, because nuclear loci take longer to sort, you don't need monophyly, you need significant divergence of allele frequencies. So allele frequencies must be significantly different. You have to do a statistical test, like a Fisher's exact test or whatever, to see whether there's a significant divergence in allele frequencies at nuclear loci. Okay, so those are the two priorities to define an ESU. Okay, so if you have a gene tree that looks like this, you have got here at least 3 ESUs. You've got ESU1, which is this monophyletic clade, ESU2, which is this monophyletic clade, and here you have another monophyletic clade, which could be ESU3. Okay, so this is a perfect definition of an ESU. Okay, if there was one blue circle here, for example, then that blue would not be monophyletic, it would be polyphyletic, and it would have incomplete lineage sorting or gene flow, and we would not be able to call it an ESU. Okay, because there's either evidence of incomplete lineage sorting, which means it's not that special because the lineages have not sorted for a long enough time, or it could have gene flow, which means it's not that special because it's in contact with other populations. Okay, it's only an ESU when it's special, when it's isolated, it's been surviving for a very long time without gene flow coming from somewhere else. Okay, that's why it is special. Okay, so Moritz's reasoning is that historical and recent gene flow, right, will make the criteria for genetic distinctiveness more concrete. So Moritz has said that if you want to say something is really genetically distinct, then it should not have had any gene flow, either historically or recently. Okay, so evidence for long-term divergence that is continuing the present, that's mitochondrial DNA. That's the mitochondrial DNA reflecting this long-term restriction of gene flow, and divergence at nuclear microsatellites, for example, significant divergence in microsatellite allele frequencies, for example, would be taken as of historically restricted gene flow, which is still restricted today. Because remember, microsatellites will give you the more recent events, whereas mitochondrial DNA will reflect events that are further back in time. Okay, so then below the ESU, there's something called the management unit. Now, the management unit is where we can't demonstrate monophyly. So either the lineages have been sorting for not long enough, that lineage sorting has not become complete, that monophyly simply has not evolved, or that monophyly did evolve, but then it got messed up by subsequent secondary contact and gene flow. Okay, so gene flow came and messed up the monophyly. So either way, monophyly cannot be demonstrated. It's either paraphyly or it's polyphyly. Okay, so but microsatellites show significant allele frequency differentiation. Okay, so when monophyly, you cannot find mitochondrial DNA monophyly, but you do still get significant differences in allele frequency at microsatellites. So what is that telling you? It's telling you that because there's no mitochondrial monophyly, it is only paraphyly and polyphyly. It's telling you that the population is not special enough to have been restricted for a long time. Gene flow was not restricted for a long time. Okay, but if microsatellites, which evolve much more quickly than mitochondrial DNA, if microsatellites tell you that there's significant differences in allele frequency, then that is telling you that recent gene flow has not occurred. Okay, but the fact that they're poly or paraphyletic and mitochondrial DNA says that, well, it could be that there was some ancient gene flow, okay, or that the divergence has not been long enough for them to have evolved monophyly. Okay, so either way, in that situation when you don't have the evolution of monophyly, or you had monophyly and it was messed up by gene flow, you cannot consider that population an ESU. Okay, because you can't demonstrate that there's been no gene flow for a long time. It's not special. Because there's been gene flow or ILS, it's not special. There's allele sharing or haplotype sharing, right? But if there's a significant difference in the frequencies at microsatellites, you could then say, okay, these don't qualify for completely separate management, but they can be managed differently within the ESU. So you could have an ESU with two different management units. So an ESU, which is significantly different at mitochondrial DNA from other ESUs, but within the ESU, you've got two populations which are, say, paraphyletically related. So there's no monophyly between those two populations inside the ESU, but if you look at their nuclear DNA, the microsatellites, there's actually significant population structuring between them. Okay? So you have recently a stop in gene flow that created some differentiation, but the gene flow has not been stopped for a long time. Okay, in other words, that's why that you don't pick it up in the monophyly of the mitochondrial DNA. So gene flow has stopped, but it stopped recently, and you got some differentiation happening, but not old differentiation. In that case, you can't give it the same priority as an ESU. Okay, you give it a lower priority and you call it a management unit. So inside each ESU you can have management units underneath each ESU. Okay, and the management units, because they have not been separated for a long time, they don't have the same priority. Okay, they have a lower priority. So if you're going to the politician and you're saying, look, we've got 2 million and we want to spend it on this species, and in this species we have one ESU and two management units, you can say, okay, we will spend half the money on the ESU and the other half we will divide between the two MUs. Because why? The management unit does not have as high a priority as the ESU does. And that is why conservation units exist, because we want to prioritize. Where should we put most of our money? Where can we put some of our money? Okay, that's why we need to have different priorities. Okay, but be that as it may, the ESU and the MU, this concept of Moritz, it still requires thorough understanding of genetic structure. So you can't just define a management unit and an ESU without doing genetics and good high quality genetics at that. So you got to sample widely, do a big phylogeographic study, looks at lots of loci, microsatellites and mitochondrial DNA, and then only can you figure out what ESUs and MUs that there are. Okay? But like all problems, like all solutions in biology, because biology is not like maths, 1 + 1 = 2, yeah, biology can be it 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 depends on how much data you have collected. Your interpretation could change quite wildly depending on how much data you analyzed in the first place. So it's always a good important to do a big study with lots of hundreds and hundreds of samples and lots of loci. But nevertheless, ESUs can also be problematic because biology cannot really be compartmentalized the way humans want to compartmentalize them. So, although the ESUs and MUs have been applied to many conservation cases, it's also been heavily criticized. It's been heavily criticized for a couple of valid reasons. Okay, for example, if we define ESUs based on mitochondrial DNA monophyly, but ecologically the two ESUs are doing exactly the same thing. So they're feeding in the very same area and they're adapted to the very same environmental conditions. That could still They could still be different ESUs. So we could still give them high priority, but some scientists might say, well, they're not adaptively different. So you're going to give the ESUs high priority, these two ESUs high priority, but they're not actually doing different things. So if one of them goes extinct, the other one's still doing the same thing, right? So we still got some animal doing that very thing. They're saying that more priority should be given to populations that are adapted to different things, where the environment is different and the animals are doing different things. That's more that you should put more emphasis on that than on simply mitochondrial DNA monophyly. So that's one criticism. ESUs also don't have a geographic component, so you don't know where in geographic space the clades are located, and it does not. That's right. It very importantly, it does not consider whether a population is able to recover. So even if you say, okay, this ESU that you go to the politician, you say, oh, we've got these, Your Honor, we've got these 3 different ESUs. We need to put 1 million on each of them. But what if one of the ESUs, there's only maybe 3 individuals left and they're all female? There's not even a male. The last male is gone. So in other words, that ESU has already inside the extinction vortex. It can't be pulled out. You cannot survive it. You cannot save it because there's no male left. Like the northern white rhino. 2 females, no male. And yet it is an ESU. So shall we keep putting 1 million into that? Why? We will never save that species because there's no male. It's already extinct. There are 2 females left. So can we justify spending 1 million? So there is another way where the ESU can be problematic, right? You can say the northern white rhino is an ESU and say to the politician, we have to save it. But the politician can say, but there's no male. How are we going to, if we can put even 10 million here, we're not going to just create another male. So we're not going to save this species. So again, the ESU, you can define it, but it may not, it does not consider whether that ESU is actually able to survive. Okay? And at that stage, I would like to leave it. We will get into that and other things a lot more in honors. I want to just repeat again, please make sure you go through all the lectures that I've so painstakingly made for you. And if you study from the lectures, take all my tips to heart, you will do very well in ZOO3649. Okay? And I know, but by now you're probably very, very tired of hearing my voice. And so I would just like to leave it here for the module for this year. And wish that if anyone has any questions, please contact me via email. We'll try to do at least 1 revision session before your exam happens. So stay tuned for posts about that revision session. And apart from that, please study hard. You have got all the materials to do well. Okay, I've given you everything you need to do well in this module. So please, now it's up to you. Okay, study hard, think hard, understand the material, and you'll be fine. And I'll see you next year for honors.